So let's move on to the last chapter, which is about the uses of polyploids in plant breeding. So, we started this course with comparing the size of diploid versus tetraploids, because indeed, to have a bigger size is of interest, obvious interest in breeding, and this is called the giga effect, which means that polyploids, like here, strawberry octoploids, are bigger than diploids, and here we saw this case already. And how this happens in terms of mechanism is very simple. Let's look at leaf cells here, comparing diploid versus tetraploids. The difference between those two is that the cells are bigger. If we take the example of here, for example, this cell is bigger than this one. And if we look at stomatal cells here, so the stomata, you see also they are quite much bigger in tetraploid compared to diploids. And that's actually just a reaction of the cell. If the nucleus is bigger, the cell will expand more precisely. And that's the case in tetraploid, of course, because you have more DNA to be contained into the nucleus. So the nuclei are usually bigger in tetraploids, like here, compared to diploids, like this one, for example. But the way that the cell does that, actually, just expand and increase or fill the volume with water. With the cytoplasm is mostly water. So in the end, the problem that is in those giga effect polyploids is that it doesn't increase the dry biomass, the molecules. It only increases moisture, which is water. And it leads in terms of taste, it leads to watery fruits, which is not a trait of interest for consumers. But then, if we look at plants that consumers don't want to eat and just want to look at, like ornamental flowers, for example, then it's pretty interesting because the flowers of polyploids are bigger than flowers of diploids, and then it's more beautiful, let's say. And another point is that polyploids usually have a longer flowering time, which is also of interest because if the flowers are here for longer, consumer is happy. Now, another interesting point is what is shown here. So we talked a lot about how bad polyploidy is for fertility, but actually this can be a good thing. In the case of watermelon here, some people don't like seeds, like those black seeds, and just want to eat the flesh of the watermelon. And that is actually performed, and what you have in the supermarket with seedless fruits is actually triploids. So they are sterile, which means they can't form viable seeds, and what you have here, this white stuff, is actually aborted seeds. So basically, no seeds, and the consumer is happy. And it's the same with banana. Actually, all the bananas that we eat nowadays are triploids, and they are propagated vegetatively. And if you look at the diploid relatives, they're actually full of seeds, and it's quite not edible. So sterility can be interesting in some cases. Now let's look at another aspect, which is inbreeding depression. So when you want to have a pure line, because this line has a certain trait, like let's say yield of the grain yield, you want this line to be homozygous, and you want that because you want it to be stable over generations, and you want to be sure, and you want to know that if you take the progeny of this line, you will still have the same trait. But that leads to other problems, which is homozygosity can be bad if deleterious recessive mutations becomes homozygous. So at the heterozygous state, you won't see any problems because the dominant normal or okay allele will be expressed. But if you have homozygous deleterious mutations, then you start to see defects in the plant, and that's what is called inbreeding depression. And an interesting thing, we talked already about how in polyploids you can have a lot more combination of alleles, and it's not only that, you also will need more time to reach a homozygous state, just because you have 4 alleles, and the probability to have them 4 together in the same individual is rare, just as a matter of probability. So that's what the curve is showing here. You have the coefficient of inbreeding, which is basically the rate of homozygosity in a plant. And you can see that to reach the same rate in a diploid versus a tetraploid takes much less time in a diploid. And it's just because of, again, probability of having homozygous alleles. So that's quite of interest, of course, if you want to mitigate like inbreeding depression effects. Now, let's go back to the Trachopogon example again. So as we discussed, several individuals, even within the same population, have different karyotypes. That probably leads to different traits, and that actually can be of interest for breeders. Imagine that you have allopolyploid progeny, and they all have very different traits. Then, from these traits, you can select whatever you like in terms of yield again, or resistance, or whatever, growth, and that's a good pool of genetic variation to start with. Then we also mentioned before synthetic polyploids. What means synthetic means that you produce polyploids and you use for that a chemical compound which is called colchicine. What is colchicine? Actually, colchicine is a mitotic inhibitor, so it disrupts microtubule polymerization, and microtubules are very important for the chromosome movement and chromosome segregation within the cell, and it actually will block the cell at metaphase, so when the chromosome pair with each other, okay? And the point is, at metaphase you still have the sister chromatids that have been produced. So basically you will have, in a diploid case, you will have 4 copies of one chromosome, because you have 2 chromosomes and each chromosome has 2 sister chromatids. And the point is, the cell will be blocked at metaphase, and instead of proceeding with the rest of the mitosis, the cell will exit mitosis, which means the cell will exit mitosis with a double amount of chromosome, which will be 4 if the cell was diploid. And then how you do that, you just apply this chemical compound on mitotic cells, more precisely meristems, and as we discussed, meristems, shoot meristems, leads to flowers and then to the next generation. So if the meristem is tetraploid, the progeny will also be tetraploid. And that's what we call synthetic polyploids. And that is actually very used in breeding, and especially in the case as represented here. So we have here bread wheat, rye, and durum wheat. The point is, bread wheat is of course very interesting in terms of grain yield, but on the other hand is not the best in terms of resistance to some pathogens or resistance to cold. But rye, which is then much less good in terms of grain yield, is quite resistant to, again, cold or pathogens. So of course the first ideas that breeders could have and had was, okay, let's just make hybrids from those, and then if we're lucky we should get traits from both that we can select for, and we will get a much better crop. So they did that in both cases, and then they end up with this hybrid. But then the problem they faced is that, oh, this F1 is actually sterile. And why is that so? Well, let's go back to what we discussed first. So if we have a hybrid between 2 species, you will have what is called amphihaploid. That means that it's not a real diploid or triploid or whatever, but instead each of the chromosomes will behave as being haploid. And the reason why is because during metaphase 1 you won't have any duplex between homeologous chromosomes. They won't pair, which means they won't separate, which can lead to unreduced gametes, but can also lead to aneuploidy of the gametes, because the chromosome won't segregate properly and then they will be sterile. So that's when the idea came to make polyploids of those. And what you do, so you just use colchicine, as I told you, and then, surprise, doubling the amount of chromosome leads to fertility again. Why this happens? It's quite simple. It's actually, in this case, as I told you, the chromosomes from genome A, B, D, and R are not able to form proper duplexes during metaphase. But now that they are actually polyploidized, chromosomes from genome A will be able to form duplexes with the other chromosome from genome A, B with B, D with D, and R with R. So in the end, this hybrid, polyploid hybrid, will actually behave as a diploid in terms of meiosis behavior. And here is the same. So in that way, breeders could get fertile hybrids, which means high grain yield from wheat and resistance to cold from rye. So both traits into one. Another interesting thing with colchicine is you can create what is called double haploids. So sometimes if you want to have a pure line, as we discussed, a very fast way is to regenerate plants from haploid material, so gametes basically. But of course the point is now if you get haploid plants, they will grow, no problem, but they will be sterile. Because of course again at meiosis, if you have a haploid genome, it's not possible to get proper segregation of chromosomes. So the simple thing to do is after you get the haploid new plant, you can just go back to the diploid state using colchicine. And that way you will have again fertile plant. But then, of course, polyploids have some pitfalls. So it's not a miracle plant. So one of those is, of course, meiosis defect, as we discussed. If we have reduced fertility or reduced or even pure sterility, the problem is that first, you won't be able to have a progeny, so you won't be able to propagate your plants, and second, you won't be able to have grains in terms of when the crop is used for the seeds, like wheat or maize. If you don't get fruits, then the crop is not useful in any situation. The other point is when you want to have pure lines, genomic instability is a bad thing for you. And then another point is when you want to create hybrids, crossing those polyploids with diploids is quite complicated. First of all, because you're going to get sterility of the progeny. Let's say if you cross a tetraploid versus a diploid, you get a triploid, and a triploid is usually sterile. Another point we won't talk much about is called the triploid block, which is if you try to cross a diploid species with a tetraploid one, the seed of those actually won't even make it for some physiological reason. And then the way to bypass that is actually to bring the polyploid back to a diploid state, which is called diploidization. And how you do that, you just cultivate, as I showed you before, you just regenerate a plant from pollen, which is called androgenesis. And finally, another point, you can, if you have enhanced heterozygosity, like what I explained to you, and with reducing breeding depression, the other way around is also true, which is you will need longer time to get pure lines in the case you want to get pure lines. So, thank you for your attention and let's meet soon.